This is designer stuff. We can predict how things are going to behave before we even get into the clinic. You know that we've had literally 0 technical surprises so far.
Bionics is an online, peer-to-peer platform for R&D stage licensing and collaborative partnerships in the biopharmaceutical industry. In other words, Bionics provides a user-friendly infrastructure for biotech and biopharmaceutical companies. This enables development stage biotech companies to display their novel compounds available for licensing on a global scale. On the other hand, the platform offers biopharmaceutical companies direct access to these innovative compounds, which will enable them to fill their R&D pipelines.
Hi everyone, my name is Simbad Rafalian and I'm the founder of Bionics. Bionics is the first open exchange platform for R&D stage licensing and collaborative partnerships in the biopharmaceutical industry. At Bionics, we work together with research institutions, biotechnology and pharmaceutical companies that are discovering and developing innovative compounds by connecting them through our online biopharma R&D exchange platform. Helping our clients to gain for their compounds a vital exposure to the right partners at the right time will increase the likelihood to get these innovative compounds into the clinic and ultimately to the patient as a novel medicine. These interviews simply are part of the Bionics vision, which is providing exposure to biotech companies and their R&D pipelines in order to find the right licensing and co-development partners without spending lots of time and money on conferences, especially in our days when there is no conferences. Today, my guest is Jeff Galvin, CEO and founder of American Gene Technologies. American Gene Technologies is a US-based biotech company developing a novel gene and cell therapeutics in large and orphan indications, infection diseases, immuno-oncology, and monogenic disorders based on its proprietary lentiviral platform. Hello, Jeff. Thank you for being today with us.
Thank you for having me.
Thank you. Can you please tell us more about American Gene Technologies and its R&D pipeline?
Sure. So American Gene Technologies was founded in 2008, and we have been developing a platform of reusable components that can be mixed and matched to cure a myriad of diseases. And we have lead programs in 3 different areas. One is infectious disease, and the lead project in that is a cure for HIV. That is already we've already filed an IND with the FDA, and we're in discussions with them to get the last couple of questions answered on that. And we hope to start the clinical trial this year. I have very high confidence that we can take chronically infected HIV patients, so HIV positive patients who are well controlled on antiretroviral therapy, we can convert them to a functional cure. They will no longer need their antiretroviral medication every day. They can never infect anybody else. They can never progress to AIDS, and they can never be reinfected. We can talk about more about that later if you're interested. Then we also have a monogenic platform that's led by a cure drug candidate for phenylketonuria. We've got data in animals now that looks promising on that. And that would be a small lentiviral injection into the liver, and that should restore the normal morphology of that disease. And we think it's a potential cure. And then finally, we have a very exciting immuno-oncology portion of our platform, where we have figured out how to stimulate your naturally surveilling cells for epithelial solid tumors. So you have 3 to 5% of your circulating T cells are gamma delta T cells that just spend their entire life looking for small malignancies and eliminating them before they grow into tumors. Well, it turns out that even if you have a tumor that has escaped that surveillance, we're able to stimulate that surveillance up to a level where it will eliminate those solid tumors. And it looks like we can do that by just treating the primary tumor. And this actually causes a body-wide clearance of not just that primary tumor, but these stimulated cells will go and clear secondary tumors and metastases as well. So very exciting stuff off of this platform. And we are essentially, we think of ourselves as a cutting edge gene and cell therapy development or pharma company. And I think what's unique about us is this whole idea of platforms where you have components of sort of genetic constructs and a whole bunch of innovations and patents surrounding the use of the viral vectors, not just the viral vectors themselves. We're viral vector agnostic, but how you use those vectors, what sort of constructs you can put together to address a wide variety of diseases. And we have a tremendous platform on that at this point.
Very interesting. And what's very, I think, exciting that you are not only going into one disease area, but you are approaching in many disease areas, from large indications to orphan indications and even immuno-oncology. And I believe personally, this is the future. When you have like a very cut-aging gene and cell therapy, you have to go to different areas, and ultimately the one that will win the finish line will be, I think, the success story for the entire company. Because, you know, oftentimes our experience showing biotechs taking a very narrow area with their entire platform technology, and oftentimes they cannot really breach the time, and they don't really have so much resources to stay in one space, correct?
Yeah, you know, I think you're absolutely right that most of the companies that have entered gene and cell therapy have done it in a very siloed way. They tend to think of the viruses themselves as being the platform, but we think of those as just the delivery mechanism for the software that goes on those. So think of them in the software world as being the diskettes, right? Well, the diskettes would be meaningless without the software on them. So lentiviruses, AAV, okay, they may have characteristics that make them appropriate for delivering things to certain parts of the body. But the key thing is, what do you do when you get in there and you tweak the DNA or the operation or the molecular biology of a cell, right? Now with lentiviruses, we're adding genes that can either upregulate any gene product or downregulate any gene product. So that's a very useful thing, that we have these diskettes, lentiviral vectors, that we can put things on where we can up or down any gene, and we have different ways to steer them to different parts of your body. We can put specific promoters on them so they only turn on in disease cells that need treatment or in specific types of tissue. And so this is a really, you know, just sort of amazing breadth of capability that we have to make highly targeted yet highly powerful therapeutics that are tweaking with the drivers of everything that goes on in your cell. Sometimes the question is like, what can't we cure with that? Because I think what you're seeing is that every year our capabilities are doubling and the cost of doing what we're doing is halving. Yeah. And it reminds you of sort of the old days of Silicon Valley, you know, Moore's Law about, you know, processing power. And that's what's going on in this industry as well. And in that environment, what's possible, right? The answer is anything. If you wait long enough in that environment, the possibilities just get to be endless. And this is what we saw in other technologies and other technologies that I've even been part of. But I think that, you know, your basic question is absolutely right. A lot of companies are making the mistake that you can use the old models with this new drug development modality, and you can be successful that way. And you can for a short period of time, because if you find anything that works and then you make $100 million bet and put it into the clinic, and if it works, you make billions. I mean, like Avexis, for instance, right? So the old style of biotech success still works, but it's not the most efficient way to do it, and it's not the long game. That's sort of like the difference between being Lotus 1, 2, 3 and being Microsoft, right? Lotus was the #1 software company on earth in the early 80s, and Microsoft was nothing. But look who's here now, and look who's gone, right? Because the bottom line is Lotus was incredibly powerful in one siloed area of spreadsheets, right? But Microsoft had this tool set. They owned the tool set. They were efficient at using the tool set. They were actually building the tool set, and they had a efficiency of putting together applications that was unmatched at any other software company. So they could pick and choose where they decided that they would be dominant in the future. And at some point, they set their sights on spreadsheets, and now Excel dominates, right? And that's the nature of this new software industry, writing software for the human computer, the human cell.
Yes.
It is that efficiency is what matters. AGT believes that we can take a standard monogenic disease, once we have the tool set, so as our tool set progresses, more and more monogenic diseases come in scope of our platform. But we believe that once we find that it's in scope, we can get from idea to first human efficacy in 2 years and $10 million. That is an unheard of efficient number. Yeah, in the world of developing drugs. But how else could you address the 7,000 monogenic diseases that will eventually be cured with viral vectors or with some form of gene and cell therapy? There aren't enough $8 billion payments in the world, you know, in order to buy out 7,000 new drugs, right? These things could be made very efficiently. We're going to license out our platform eventually once we prove that it works. Then we'll go ahead and collaborate with other companies that are interested and have knowledge in specific siloed diseases. But in the meantime, we're going to prove that with HIV, phenylketonuria, and cancer.
Great. So it seems like a very promising pipeline and interesting way as you approach the industry. But it brings to the next question naturally, which is like you touched to the point, licensing and collaborative partnerships. So what do you think, what's your opinion on interest in R&D stage licensing and collaborative partnerships?
Well, right now we're talking quite seriously about out-licensing our PKU therapeutic. And the reason is that we have enough data on it in animal models where some of the more forward-thinking pharma companies are getting interested in maybe partnering or in-licensing it. So I think that that may be one of the first things that goes into play. But HIV, we've been trying to partner that for a while, but the very big pharma companies that are really calcified in the old models want to see first human efficacy before they can put it into their basic, partnering or business development models, right? Now, it's sort of you snooze, you lose situation, right? Because if we show first human efficacy in HIV cure, well, we can just go public and become the new Gilead, right? Meanwhile, Gilead has an opportunity right now to partner with us. And of course, they're going a little bit earlier than they like to, but it would be sensible if they had that kind of flexibility to partner earlier and to either take that risk, which is a very small number. Remember, we're not talking about a ton of money to develop this product. So it's not a giant risk for them, but to be there on day one when this thing shows first human efficacy and be able to grab it up. Because Gilead could very easily become the new Gilead, right? Where they don't just treat HIV anymore, they treat you for 1 to 3 years on antiretroviral therapies, and then they cure you. And what's that going to do to the antiretroviral therapy market? It will all go 100% to Gilead, right?
Yes.
It could also go to J&J or ViiV, right? So it's going to be a whole new world in the pharma industry. Things happen fast in gene and cell therapy. Some of these people are going to be caught, sort of asleep at the wheel or, back on their heels a little bit. And it's going to be very interesting to see. I think this is, the whole gene and cell therapy area is highly disruptive to the existing pharmaceutical industry in a way that benefits patients, society, insurance companies, and even the pharma industry, because, the reality is that it's a $10 trillion healthcare market, but it could be $20 trillion if we could cure a lot more diseases. So there's lots of money to be made for pharma companies, but they're going to have to figure out how to get into this market in a serious way. And the old models of just buying up successful products may not work the way that they used to. There's going to be too many successful products. This isn't just a new drug development modality like monoclonal antibodies that turns out, 5 new drugs a year or something like that, the existing pharma companies can just divide them up between themselves strategically. No, what will happen is you're going to start to see thousands of drugs coming out of this. There are 7,000 monogenic diseases that will eventually be cured. Our cure for HIV works on hepatitis B, HTLV, human papillomavirus, herpes. We can go ahead and change very small things on that part of the platform and do additional diseases. You know, this is going to happen much faster than pharma companies are used to.
And absolutely, and this is exactly this interview is about, to bring the message, the right message to the potential partners, because once they understand the unique proposal that you give to the industry, they are going to compete for American gene technology. All companies, I mean, as you are saying, it should be very early stage. Otherwise, if the product will be ready and it will be already human data, of course, it's easy to come and to put lots of money on the table. But the question is, who is this company that understanding the technology in a very early stage? So I think this is exactly the moment that I give the right message to them.
Yeah, and I think, you know, to simplify it this way is this. The question will be, can pharma companies get their money in ahead of Wall Street when these new drugs appear, right? Because you can definitely see that HIV could be a company on its own. You know, there's enough people in the world with HIV that's got decades of life to it. And, you know, something that might even cost as much as $15 billion to acquire early could turn into a $200 billion company on its own, because you're talking about just a huge amount of not just the revenue that exists, the $32 billion a year in antiretroviral therapy. Those patients who are being treated on that have about 3 to 4 times more money spent in treatments that are associated with either the toxic side effects of the treatment or the long-term consequences of a low-grade chronic viremia. And so the money that can be captured into a pharmaceutical category like HIV is substantially larger than what it is today, because there is much more value to be generated than just extending lives for a certain period of time. We can increase the quality of life, which has huge value to the patient, and we can decrease the spending on all sorts of treatments that are only required because people are on long-term toxic chemotherapy, antiretroviral therapy. So this is a very exciting area. It is worth moving early on. We're out talking to everybody. We don't see a lot of people getting it in the HIV space, but you know, the good news is Wall Street's lining up as well. You know, they're being crystal clear that a first human efficacy data that gets public attention, and I don't know how it wouldn't, right? If you were to cure, you know, a dozen patients on something that was completely practical and safe, would that get a lot of attention? I think so. So they feel like, okay, if you get that day, we can just take you public. You don't need anything else. You don't need to out-license it or anything. So, you know, there's a lot of ways to sort of skin this cat and to get these technologies out to the public. That's our commitment. You know, the organizing principle of AGT, our mission has always been to efficiently leverage the power of gene and cell therapy in order to reduce suffering and reduce early death from serious human diseases for the most patients possible, the quickest possible. And you know, you notice in there, it's not a profit-oriented thing. Of course, there's profit in that mission, but it is really focused on efficiency is number one. And number two is to just be thinking about what solutions look like and thinking back to our platform, picking out those things that are now low-hanging fruit that look very reachable, and to start a project to get to that point. And then we worry about getting it to the public later, because there'll always be a market for a cure. And finding the patients won't be that difficult. So the pharma companies have to think about, well, where are they going to sit in that value chain? And if they don't move earlier, that's going to be a problem.
Yeah. So the early mover will win in this situation.
Yeah, the early bird will get the worm. as they say. I don't know if you've heard that expression.
So let's touch a little bit the other side. What co-development partnerships have you developed until now to accelerate discovery with biopharma companies and research institutions with your?
So most of our partnerships are in the CRO, CMO area. So we have purchased a lot of manufacturing services in the area of cell processing and viral vector manufacturing. And this is one of the nice things about this industry is you don't really have to have your own manufacturing, which can be very expensive to build and maintain. You can instead share a facility at a CMO. You can get a slot in that. And although it's way more expensive on a per milliliter basis, it's way cheaper on a per annum basis. And so it's a great opportunity for CMOs to make a lot of money opening up these production facilities. And it's still very cost effective. And it allows us to focus on our core competencies, which is really designing the elements that go on the viral vector and designing the actual vector plasmids and stuff like that, not doing the actual GMP manufacturing. So we also contract a lot of CROs that make FDA compliant assays like release assays and things like that. And so, you know, that's really where most of our partnerships have been. We have a lot of discussions with pharma companies. So pharma companies regularly meet with us at shows, and now they're doing it online during this COVID crisis, which is why I'm dressed so informally. My apologies. And but they're getting updated on a regular basis about our progress. I think they're, you know, waiting for first human efficacy, but, you know, we're trying to reach out and we're trying to educate the pharmaceutical industry and all the pharma companies and to engage in partnerships. And I think it's just a matter of time. You know, I think that the trajectory of those companies is to go earlier. There are some companies that are making huge financial commitments in that area, but I don't know that they're necessarily super smart about picking things at sort of cost-effective points for them. But I think that, you know, there's no question that everybody's getting smarter now that this technology has been around for longer, and there's more examples of things that have worked and haven't worked, and there's more expertise out there in terms of being able to get a gut feel of promising projects even before you see first human efficacy. So we're good at spotting all that stuff. If we don't end up you know, out licensing a lot of products between now and you know, when we become a public company, then we'll end up in licensing a lot of stuff because we know how to spot a winner.
Yes.
Of a project. We know how to look at the preclinical data and figure out, okay, what is likely to work, because we do it internally all the time.
You have already the eagle eye, right? So you can watch from far and really have the holistic view about that. But then can you a little bit be more specific in terms of like American Gene Technology, what kind of collaborative and licensing partnerships is seeking, if it's seeking at the moment, whether it's exactly for the product candidates in the pipeline or it's for the platform technology, so our potential partners on the other side can understand that's more precise.
Sure. Okay. So the PKU asset is being shopped around as we speak. There is some initial I think serious interest in that. But if there's a pharma company out there that wants to acquire a fully developed plasmid system that creates viral particles that can be put into the liver of a adult human or even a neonate or a toddler that restores normal phenylalanine hydroxylase and mitigates the disease for life, a one-and-done treatment based on lentiviral particles. We have preclinical data that's quite convincing in that area, and that project is really ready to go to a pre-IND meeting, and they could go with us to that pre-IND meeting. We can probably get it to first human efficacy with proper funding by the end of next year. So that's getting close. The HIV is even closer. We are certainly open to partnerships already on that. And there's a lot of ways to start partnerships that maybe start off with baby steps, or somebody may look at the preclinical data and they may feel the same way that the who's who of HIV in the world has felt when they looked at our preclinical data, that this is going to work, and they want to, jump in with both feet. We're open to, something that sort of ratchets up, or, something that is, they want to take it and develop it on their own on HIV. Now, that's not a cheap one. That's much more expensive than PKU because the market opportunity is so much larger and the IND is already filed. But, we're still looking for partners in that. And so, we're entertaining all inbounds from pharma companies, and we're reaching out to pharma companies that we know look at infectious disease or HIV as a strategic area. On the immuno-oncology, it's so big, it's hard to value at this point. I think that ultimately, if the immuno-oncology asset is as good as it seems to be showing in mouse studies, we're getting 85% complete remission in animals with one treatment to the primary tumor. You're getting abscopal effect all over the mouse's body, and it is a genetic solution based on human gamma delta T cells, and then we're doing this against human cancer. So this is likely to translate well to humans. But 85% complete remission with one shot of viral vector with our proprietary cargo. And so I think that it works for all epithelial cancers, so breast, prostate, lung, liver, colon, kidney, ovarian, pancreatic, head and neck, and skin cancers, and others. It's going to take a little bit longer to get it into the clinic. Our partner is Stanford Medical School for developing it to the clinic. But I think that's $1 trillion market at some point. There's 900 people a day dying of those types of solid tumors in the United States alone. When they die, it costs about $1 million to the healthcare system for them to die. We can probably cure them for less than half that price. Think about the upside, not just that they get their life back, but you save $500,000 of patient costs, and they go out and start paying taxes and insurance premiums again, right? I mean, so this is a win-win for everybody. But, we'll have to see on the immuno-oncology. We're certainly accepting partnerships, but it's not really sort of a licensing discussion at this point on that asset. It's too hard to value right now, but people can go ahead and get their fingers into this by either investing in AGT. We have openings for companies to go ahead and form partnerships that are sort of investments in the company as a whole or investments in particular products. So, there are ways to partner on that. And then the last thing is really any infectious chronic viral infection. We can start partnering discussions on that. It's much earlier stage, so we have really pre-clinical data on things like HTLV and hepatitis B. And pick your monogenic disease, and we can do the thought experiment. Knowing our platform, we can literally give you a probability of whether we can cure it. We can get to a go, no-go decision in a tiny amount of money. I mean, pocket change for even a emerging company to spend. And then we can get to first human efficacy with internal costs that are somewhere around the range of $10 to $20 million. And, there's very few monogenic diseases that wouldn't be profitable if the development costs were, kept under the $100 million to get it to market, right? And I think that is highly likely with lots of go, no-go decisions in the middle that, conserve capital and don't end up throwing money into a black hole. And that's the normal development process around AGT. So if somebody came to us with a project and wanted to know, what can we do therapeutically or curatively for a particular monogenic disease, we could probably answer that question, on a CDA. And we could, give them reagents to test at so little money it would make their head spin.
Very good. Very impressive data that you already have until now at American Gene Technology. So we have another 4 minutes to end our amazing, very interesting interview. But I would have another question to you. Could you give a little bit more understanding what separates American Gene Technology from other biopharma gene and cell therapy companies? So what's the main competitiveness? I think you already give good understanding. And then last question would be, what you would tell to Bionics audience that probably you want to speak out, because there might be lots of potential investors looking for probably your IPO, if so. On that, so please go ahead and tell about that as well.
So I think the specialness about AGT is that we play the long game in this industry. It really does boil down to that metaphor that I told you about, the difference between Microsoft and more siloed companies like Lotus or Software Publisher or whatever. We really are talking about the ACTGs of DNA that we can now play around with. And that's like the zeros and ones of your personal computer. And so this is a software model. It's a layered model. You have the people that make the transport layer, the diskette manufacturers, the lentiviruses, the AAV, the murine retroviruses, and who knows what will come next. Then you have the people that have tools and reusable components. That is our area. And then you have people that are building apps, right? But a lot of people are bypassing the tools layer, and they're building right on the viral vector. Now, there's nothing wrong with putting the bits and bytes right into the viral vector, but without that tool set, you can never be really efficient at doing it. So that's the secret sauce, the specialness about AGT. It's that we looked back at the software industry where I come from, and we said, what is the pivotal layer in software that allows you to play the long game and to be a long-term player in this industry? And we looked, and I decided that that is the tools and reusable components, or what you would commonly call the operating system or the platform. And that there's been a big misunderstanding in this industry so far about what a platform is, because they're used to things like a monoclonal antibody platform is something that spits out 10,000 random molecules, and then you screen them down to 1 or 2 worth testing in a mouse. That's a platform, right? But you don't have anything until something actually gets into at least animal experiments and shows a lot of promise, right? So you're $10 million in 2 years in, and all you've got is 1 or 2 molecules that are almost like a random discovery, right? Yeah. This is designer stuff. We can predict how things are going to behave before we even get into the clinic. You know that we've had literally 0 technical surprises so far on the HIV project, that we were able to test things in the cell culture hood, anticipate the stumbling blocks and the landmines of the clinical path and the development of the cell process and all those things, and do that at a very, very low cost because patients weren't involved in the process of debugging our software. We could test it in models that we've developed, and then we refined it. By the time we actually went into doing the cell processing development, it literally went according to plan. So this is a whole new, style of drug development that doesn't match anything else. I haven't seen competition that is looking at this new drug development modality in the same way that we are. So I think as the Microsoft of this versus Lotus, I haven't seen anybody else that sort of made that distinction yet. You could also look at all of our individual products. You could look at something like HIV, which I think that we are 3 to 5 years in the lead beyond everybody else. And we're also already working on the next version of it, because that's another reality of software. And that is that your first version won't be your last version. And if you don't keep yourself on the cutting edge, somebody else will come in and find another way to do the same thing. And if they're better, faster, cheaper, then they're going to steal your market. This will actually be a competitive pharmaceutical market, just like software has remained competitive. Even Microsoft, dominating different areas, can't raise their price up infinitely because there are other alternatives to Excel. You could go to Google Docs, you could go to Linux or whatever. There will always be competition in this new pharmaceutical industry. And what I tell our guys is it's not enough to cure HIV. You need to go back to the lab and cure it better, faster, cheaper. Because if we don't obsolete ourselves, somebody else will do it for us. And that's what we plan to do. So, we're going to develop initial drugs. I think PKU there's been articles that show the way that we're doing it is the best in class approach to a PKU cure. So I think we got a lead there as well. And then the immuno-oncology asset, it's going to take a little time before we have all of the data that is convincing to the outside world. But I got to say that we have a really good feeling with the level of efficacy that we're seeing in animal models and the theoretical ability to sort of map this to the human biology, that this is going to be very effective. And again, we're still looking at other ways to be able to ratchet up and, believe it or not, down the efficacy of it. Because you know what happens to the 15% of mice that don't get cured? It looks like they're dying from tumor lysase disease. The tumors are breaking down so fast, it's poisoning the mouse body. But remember, a mouse tumor is like the size of the mouse's head.
Yeah.
So, it may not be a problem in humans, but we're working on cargo elements that either augment or actually limit the effect of the existing cargo element that we're testing. But that's what this is all about, is that, these are designable things that react together in a predictable manner that can be tested in cell models and cell culture hoods and cheaply in animals, and that have very good translation to human beings because of the basic genetic design of these drugs versus the chemical design that operates on the outside of cells. We've got a genetic approach that operates right on specific genes. It is literally tweaking one particular pathway inside the cell. That's much more predictable than a chemical that attaches to a receptor or that permeates the cell, or
with lots of side effects.
Yeah, the side effects are so much more limited. Anyway, so I think that, I think we've got a very good competitive advantage in many different areas. We're very excited about partnering with pharma companies as they start to get more confidence in the gene and cell therapy area and understand it better and are better at picking promising projects that are still in the preclinical phase. And also, ultimately, I don't think we're really prepared right now to out-license our platform to any company that comes along, but any company that has an interest within a particular monogenic disease and has good knowledge of that disease, maybe has some animal models from previous work or cell models from previous work that would be help to be provative of a reagent that we could design for them, and they have money to fund that project, it's not too early for companies like that to reach out to us as well. So they need to be thinking, to get to first human efficacy, $10 to $20 million of our costs. So there's going to have to be some overheads on top of that. But we're still talking about something pretty cheap and that has a lot of go, no go opportunities along the way. So I wouldn't discourage anybody who's thinking along those lines. And then finally, of course, we're not allowed to make a solicitation. So this is not a solicitation for investment, but we are a private company. Accredited investors are where we get all of our money. And so we're in we raise preferred equity rounds from time to time. So there's that possibility for companies or individuals to get involved with us if they conform to the rules of private company investment.
Are you looking at the moment private investors more, or what's your investment strategy in the next 1-2 years?
So I'll tell you that we got a lot of momentum in this area before VCs started to get interested in gene and cell therapy. Remember, we were founded in 2008, and we had a lot of good stuff by 2014, 15, when a lot of VC companies started jumping in. And so we were too expensive for them at that point. We didn't fit their model where they put down, one relatively small investment for 35% of the company. We were raising money from individuals at 45 million pre-money valuation before VCs even talked to us. By the time, they started to get serious, we were at 120 million pre. And, we've now raised a total of over $40 million from mostly high net worth individuals, boutique VCs, and angel funds and angels. So, I would say that that's still our primary target, although now we're starting to get investments from family funds, hedge funds, larger private investors, the sort of billionaire class. So, this is in a world where the equities market, the public equities market is getting a little bit volatile. This could be a value play for some of those bigger money people where they can say, okay, we know exactly what we're buying here. They have an intellectual portfolio that represents the net asset value of the company that can be evaluated. We actually have a valuation of that from a credible source of $2.4 billion. So, it can't be sold for that in a fire sale, but even if the company went out of business, there is recoverable value. But then on the high side, if we cure HIV, the sky's the limit. If we go on and cure cancer after that, it doesn't matter whether it's an up market or a down market. People still get sick. Pharma companies still have money. So, that market tends to still reward value creation, regardless of what the current market psychology is on the Dow or the NASDAQ or, in the public markets. So, we think it's a great investment that way. There are some companies that are laying down money. There are individuals, and we're money's green. It buys lentiviral GMP production. Everything else around here is pretty cheap to run, and so we have a highly efficient organization and lots of opportunities at a reasonable price, and those opportunities are available to people that match the required criteria of the SEC for investing and talking to us about private investments.
The company is so interesting. I guess some of the our Bionics audience would be also interested to invest as a retail investors. So is there any way that we as a retail investors could expect anytime American Gene Technology going IPO?
Well, that's one of the options that we are pursuing. So it may be that in the early years, we will do something like transact the HIV product, and that will put so much money in the bank that it would be better for us to stay nimble and be out of the public market so that we have more control over our destiny and less sort of resistance against the wheel. Because when technologies are on a rapid climb, you want to be able to grab every brass ring that you can. But the other thing that could happen to the HIV is that we spin it out into a separate company and take that public and keep the rest of our development efforts still in the private arena. That may be a really great opportunity that may happen on first human efficacy. So absolutely, keep your eyes out. Go to our website, connect to us on social media. You definitely want to get our newsletter, and you'll get early information about everything that we're thinking and everything that we're doing, and everything that we're allowed to disclose publicly we put on the website, or we put in our social media, or we send out in our newsletters. So we're happy to have anybody sign up for those and to stay in touch with us.
Great, thank you, Jeff. Is there anything else that I forget to ask you that you would like to let the audience know? The last word.
No, I think I really appreciate this opportunity to speak with you today, and I really enjoyed this interview. So thank you for having me, and let's stay in touch. If you ever want to come back and talk again, please make sure you get in touch with me right away.
Thank you, Jeff. Thank you for providing such a helpful information, and thank you for being our guest today. I want to remind you guys once again that our guest was CEO and founder of American Gene Technologies, Jeff Galvin. Thank you, Jeff. And if you like this interview, please subscribe to Bionics YouTube channel and follow Bionics on LinkedIn and Twitter. And don't miss our new upcoming video interviews, as well as go ahead and, as Jeff mentioned, subscribe to American Gene Technologies social media channels. So stay in touch, and we will come again with a new interview with one of other CEO of the biotech. Thank you very much. Thank you, Jeff.